Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing you another really fascinating guest involved in creating a better tomorrow for all of us, working in the fascinating area of aging, longevity, health span. Today, we have the honor of being joined by Dr. Marco Quarta, who is the co-founder and chief executive officer of Rubedo Life Sciences, a really novel biopharmaceutical company that's focused on developing a broad portfolio of innovative therapies engineered to help target cells which drive chronic age-related diseases. And their proprietary alembic drug discovery platform has been involved in engineering these novel first-in-class small molecules to ultimately selectively target senescent cells, which, as we've discussed on previous shows, play a key role in the progression of many different types of diseases, from pulmonary to dermatological, oncology, neurodegeneration, just to name a few. Dr. Quarta received his doctorate degree in biotech from the University of Bologna, moved on to do his Ph.D. in neuroscience at the University of Padua, and then completed postdoc work in aging and stem cell biology in Professor Tom Rando's group at Stanford, continued his work at Stanford, directing a research team at the Center for Tissue Regeneration, Repair, and Restoration at the VA hospital in Palo Alto. And there he helped establish their translational program in regenerative medicine and has been responsible for dozens of peer-reviewed publications and patents in the field of aging, stem cells, regenerative medicine, rejuvenation. In addition to all that, Dr. Quarta has previously been responsible for co-founding various companies, including Wetware Concepts, the Young European Biotech Network, as well as Turn Biotechnology, an organization focused on cellular reprogramming, and served as the executive board member of the European Federation of Biotechnologies. Currently sits on the advisory board for the Calpoly Institute of Regenerative Medicine, or CIRM, Calpoly Bridge program, on the advisory board of the Center for Healthcare Innovation, and is a member of the Paul F. Glenn Center for Biology of Aging Studies at Stanford. And then one last title that was just added to his dossier: he is now also the CEO and president of the board of directors of the Fadon Institute, which is an interesting think tank that is working on discussing effective and sustainable growth strategies in the aging longevity sciences space. We're honored to have him with us today. Really excited, a lot of interesting topics. But Dr. Marco Quarta, thank you so much for taking the time to come on the show.
Well, thank you so much for having me and for the great introduction. You know more about myself than I do.
Well, it's a really fascinating background, a lot that you've been involved in, Marco. And, I would really like to start things off by, giving you the floor for a little bit, just to talk about the beginning, because, reading about you, you started off in a lab run by a Nobel laureate that was involved in, discovering nerve growth factor, move on to do your PhD and, this really interesting area of myogenesis and muscle regeneration, rejuvenation. Talk a little bit about the beginnings here, because it is a very interesting set of stories that leads into the current really interesting story, what you're doing now. But take us back to the beginning, if you would.
Okay, well, that's happy to dig into my past. Well, the very beginning, and sometimes I mention this, but the very beginning starts much earlier than that, right? My inspiration about studying aging, or really naively saying cure aging, I say naively because I decided this when I was 5 years old.
Nice.
I was very young, and I even remember that day when happened and what inspired me. But that day I have this idea, right, that different organisms, come to the realization that different organisms, different living forms live in different rates. And I thought it was obvious that then that's the plastic phenomenon and that can be changed. And so I remember going to my mom and I can say, Mom, I decided what I want to be when I grow up. I said, yeah, well, if I want to be a scientist, and we have several scientists in the family and physicians. So she said, oh, great. I'm glad that you're inspired to cure aging. And she said, okay, well, good luck with that. But we can help you. And so at the age of 6, I had my first little lab in my room. I got, pieces for a chemistry kit and a real microscope, not a toy, real microscope from my uncle. Nice. And, anatomy model. And I started to play with science. Actually, some of those pieces I can still carry with me. This is from the original chemistry kit that I had here. It is in my office. Nice. And I'm still playing with it, so I never stopped. In reality, it was not a moment. And I continue. So to your point, the choice of my studies was always looking for someone I can learn from about aging, but also from a translational standpoint, how to change this. Initially my intention was starting in the med school, but in Italy there was this that it was the first year at the University of Bologna, the first degree in biotechnology. Mm-hmm. And I thought, that's what I want to do. That's way more applied. I don't want to be a physician. I want to cure the patients of the future, not the one right now. And so I went to biotechnology. I was actually the number 17th in Italy that enrolled in the program, and then expanded all over, and every university has a class. But that was the very first year. And so to your point, yeah, I studied with many people, and especially when I joined this lab that was under the supervision of Rita Levi-Montalcini, the Nobel laureate who won the Nobel Prize for the discovery of the NGF. But that lab was working on brain aging and brain degeneration, and most important for me also, how to apply regenerative strategies for the brain. So for me, it's great. I can learn how the brain works, but also how that can be changed. And that led me then to my PhD in neuroscience, where in reality I started to keep working on the brain, but I discovered also the skeletal muscle as a very fascinating tissue in many contexts, in disease, in aging, and also from a bioengineering standpoint. So there are a lot of things that you can do applied. So my very first company that I started back then, during my PhD, is applying was a business plan competition that was in Italy. It was called Start Cup. And I won it. So they gave me money to start a company, and I had no idea, of course, what to do and how to do it. But that came with angel, not investors, but also angel mentors. Mm-hmm. So that was very good for me to learn to do that. But the idea was to apply some of my science into applications and developing this kind of hybrid biosensors combining either neurons or muscle cells into microchips. Right. That can be used as a biosensors or for implantation, for regenerative medicine. That was how I started to kind of think this through in a more applied way. And that led me then to Stanford. Some of that work, working with Tom Rando, our connection, of course, was on the skeletal muscle, since it's one of the focus of this lab. Mm-hmm. And of course, being one of the leading labs in aging and rejuvenation, with this idea of the parabiosis that was launched with this lab, right? The idea that you can put together young and old mice, and they discover, they just published their work in 2025 on Nature, that by Irina Comboy, that the young blood can somehow biologically rejuvenate to certain aspects of the old mouse. And so that was perfect lab for me. Mm-hmm. And my initial idea was actually not to stay. I moved to Stanford as a visiting scholar, invited by Tom, with my idea to stay 1 year and come back. And after 1 week, I decided that I would stay. So I never actually returned, and that's why I did my postdoc and then continued to when I started my group, and that's my career followed. And that was a perfect environment because, of course, Tom and all the work around really exposed me to the leading edge of the world of aging science, but also rejuvenation and regeneration, which was exactly my sweet spot where I wanted to be. So that was very conducive then years later to start companies like Rubedo Life Sciences or Turn Biotechnologies.
Mm-hmm. It's there's a really, as you're telling that story, there's a really interesting theme that sort of flows through your publication history in the sense of you're not afraid to you weren't afraid to sort of focus on the niche, let's say, whether we're talking about sort of when it comes to say muscle rejuvenation as the example, because you're publishing on everything, right? You're publishing on sort of the signaling factors that go on in terms of sort of the myogenesis. You mentioned the sort of the semiconductor muscle technology. You're publishing on sort of inflammatory factors that affect wound healing and regeneration. And then obviously this, as we're talking about Tom Rando's lab, this sort of comes to sort of the fruition when you obviously talk about the muscle stem cell niche and ultimately how we awaken that. And you introduce, we'll get into senescence in a little bit, but you publish a lot on the topic of quiescence, that we have not just sickly cells that may be there, but cells that may be sleeping or quiet, or a lot of really interesting dynamics occurring there. And I found this very inspiring with your work, that typically we see a lot of researchers focusing on one thing, but you understood the areas. Hey, there's a lot happening here that goes into the rejuvenation puzzle. Talk about that a little bit, because I think this is very important as it leads into a little of your strategy with sort of Senescence 2.0 with Rubedo. But talk about that for a few minutes, if you would. I think it's a very important part of what you've been up to.
Oh, first of all, thank you. This is a great question. Rarely people look into my scientific work like that. It's absolutely true. And I think that, again, going back to my story, somewhat, and as the roots in my view of what aging biology is, and certainly I spent a long time thinking about it. And the more I do it, the more I realize how complex and little we know, even today. We're really scratching the surfaces. So, for example, I'm not a big fan of modeling aging. Like, the hallmarks of aging is highly reductionist. And bias. So I think you need to spend lots of time to try to, like, to your point, just put together pieces of this puzzle and kind of connect the dots. And the more you do, the more the level of complexity that emerges is clear. So definitely I have always been inspired to explore and dive deep as I can in different areas that I see are connected and help me to make sense to others. So for me, it was not jumping fields. It's never been. It's always been part of one big picture.
Yep.
And sometimes that requires me to spend years to work into an aspect. Like, for example, I realized how important is the biology of cellular quiescence, which is really pivotal in this, let's say, checkpoint where a cell needs to decide what to do, either a progenitor that is there waiting to be awakened and start a regenerative process or self-renew itself, so kind of regenerate itself, or instead moving into a more like an aging phenotype and become potentially senescent, which is a different state of quiescence, but irreversible to a certain extent. But that was, I think, one of the very key aspects, and that's why I spent years to really understand and try to master that part. It really gave me the opportunity to have a foundation that is very important for me in the context of aging, understanding the microenvironment, so the niche where cells are sitting, and learn that it's not just about a cell, but it's about the entire tissue, about the 3D and the entire environment. And it's not just about signaling, so molecules that might be shared, but it's about, for example, even the mechanical signaling, like the stiffness and of a tissue inform what the cells should do through a specific mechanotransduction pathway specifically. So there are ways the cells can sense how the environment is, even the geometry of the environment around the cell tells which genes eventually might be turned on or off inside the cell. And that's super important in the context of aging and how this process is dynamically changing over time. The different explorations for me were only building for me the foundation to give me confidence in how address, at least to a certain aspect, and I'm aware this is a still limited approach to aging, but at least to have enough confidence to know what I'm doing, hopefully in a way that I can translate into therapeutic.
Mm-hmm. Mm-hmm. I appreciate you laying that out, and thank you for answering that, because I thought there was an elegance to what you've been doing, and I encourage everybody watching and listening to this episode to check out Dr. Quarta's PubMed publications in this whole area of artificial microenvironments and stem cell quiescence. It's really interesting stuff. But let's get to Rubedo, because again, I think you translate what you were just talking about again to the what I'll call the Xenotherapeutics 2.0, and we've had the honor, we profiled Judy Campisi about a year ago, talking about sort of the history of this space. And again, as you were just mentioning, this is senescence is more than just a cell. Just like rejuvenation is more just about a cell. And maybe there's been amazing things happening in senescence 1.0, but you've really taken it to a new level with Rubedo, understanding there's different tissue types here, different mechanisms. Looking at the portfolio, you're not just in the senolytic space, but in the senomorphic thinking as well. Introduce us a bit to the company, a little of the background, and then take us into the alembic system. It is artificial intelligence, drug discovery, but really highly focused on this really differentiated nature of senescence across different tissues. Take some time and walk us through this, Marco. I think it's very important.
Okay, no, with pleasure. Rubedo, and maybe just I can make a comment about the name. And sometimes people ask me, why Rubedo, life sciences, and why? What has anything to do with aging or rejuvenation? So I took the name Rubedo from alchemy, and in alchemy, Rubedo is the last step or the last phase of what is called in Latin the magnum opus, which is a great operation of alchemy to realize and manufacture literally the universal medicine or the elixir, which is a medicine that cures all diseases and including aging. So I thought it was appropriate. And all the names at Rubedo are taken from alchemy, like alembic, which we'll talk later, is actually the apparatus that is used by the alchemists to make, during the Rubedo phase, the elixir, the universal medicine. Anyway, and so we kind of joke, and all the jokes are within, alchemic names, and we have code names like that.
I think it's really cool.
Thanks. And by the way, alchemy, I also did it because it has been an early inspiration of mine, again, in my exploration of what, aging means in human history. I realized from a, let's say, philosophical standpoint, human has been, of course, looking for a fountain of youth since ever, right? Since we know. And the alchemic literature, goes back thousands of years across all countries, Europe, India, Africa, China, etc. And at the end of the day, it's really one common theme that has been conserved and preserved, and is an experimental philosophy. People have been actually doing experiments in science, which was the precursor of modern science, although there was more to that. There was this philosophical exploration of nature. But sometimes they say things that are very meaningful and clever, even today, things that maybe we don't think anymore, but maybe they were like in the Persian Empire, philosophers were exploring. So I always found alchemy as a sort of inspiration for my science over the years. But let's go to the company Rubedo. So the idea there was initially to understand and find pathologic cells that are aberrant, emerging with the biological aging process. And it's really the end result of what we call aging. And we talk about biological aging because it has to be clear. Now, there's nothing you can do about chronological aging, right? That's, unless we can change time, at least there's not anytime soon. But there is a lot we can do about the biological aging process.
Right.
But this is not tied necessarily with the chronological age. There is common perception that, the older you are with your chronological age, the more biological age you are. But sometimes this can happen way sooner in life, right? You can be 20s and have a biologically aged skin and therefore develop chronic skin diseases of the skin. And you may need this sort of like anti-aging longevity. I don't know if we have a right name yet to call it, because those are very, maybe diluted terms. But therapies that target this type of biology at any point in life, right? We saw with COVID, like SARS-CoV-2, prematurely age certain tissues. And so we do see people coming in the clinics with interstitial lung diseases 20 years sooner, for example, that they had severe COVID. Cancer survivors, even pediatric cancer survivors, unfortunately, children with leukemia that undergoes to, chemo, radiotherapies, and standard of care, very aggressive, can kill maybe the cancer cells, but is damaging so many cells and induce the premature senescence response. We see those people have a change in their quality of life, the baseline changes, and they experience age-related diseases in their 30s, so 30 years sooner than the average person. So that's a biology that we're talking about. And with Rubedo, the idea was, okay, no matter what causes that, and that's where the complexity comes, but, we could maybe simplify aging as a result of, a complex, a convergence of errors and damage and noise at a certain level. But the result is this leads to cells to become dysfunctional, aberrant. And its obvious example are cancer cells, right? That they emerge from damage. It could be seen as a result of a form of aging, biological aging, in many ways. If it could be very early, but because you might have a genetic predisposition that damages your cell very early, so as a result can become a cancer cells. But there are many others. And senescent cells as an example, but there are can become profibrotic, can be a dysfunctional stem cell.
Yep.
So that's what we see in age-related chronic diseases. There is an accumulation of pathologic cells that are behind and driving the progression of this degenerative process. So what Rubedo want to do is to find those cells. What are those cells in a specific disease or tissue in patient populations that are, as a result of this biological aging process, causing the disease, and classifying them and find what are the right targets. So the right pathways, the right metabolic activities, all the building blocks that then we need to translate this into a chemistry that to make small molecules and engineering them in a way that can target those cells. So that's what we do at Rubedo, and we do this using two platforms. One is alembic, which is, and we can talk more about that, but is a computational strategy at this point, fairly complex and sophisticated, that include bioinformatics and chemoinformatics. And to really do that, so starting with clinical samples, seeing, do we find those aberrant cells, what's special about them, and what can we learn, how to target them. And then we translate this into a chemistry to make novel molecules, first in class, new target that we discovered, and engineering those molecules with different modalities, but in a way that can selectively target or be activated by your target cells that you initially discovered. So closing this loop. And why this selectivity is so important for us? Well, because if we think about going after, these are not low-hanging fruits.
No.
These diseases, it's something that medicine has been probably procrastinating or not going after for the past century because it was harder. Now technology and knowledge is better, but there are still harder conditions compared to other indications. So the bar is higher. The safety, these are talking about people that have comorbidities often, frailty, and different metabolism. So you need to make sure that you make drugs that are tolerated, especially in the context of chronic diseases that might need to take this for a long time, maybe in cycles, but still, or for me, for the rest of their life, to manage or to prevent a disease to degenerate. So safety profile, very important, and selectivity, because we want to avoid the chemotherapy effect. Then, it's not killing something that grows quickly. So I can, like an oncologist, their priority is to keep you alive now for the next 5 years. We want to keep you alive for the next 50. So we need drugs that can very selectively target what you want and prevent off-target effects that can lead to secondary problems. And as a matter of fact, you put a patch on one way and then you create a damage somewhere else. As we know, all those cellular stress and damage over time, they only cause biological aging in one form or the other. So that's really the goal is to make molecules that are highly selective and targeted, which is not that easy. So we really build a platform and technologies to do that. So how we did that? So you ask about the alembic. So alembic is leveraging single-cell multiomics in multiple ways, from spatial transcriptomics or multiomics, single-cell RNA sequencing, ATAC sequencing, metabolomics, etc. So lots of data that we use and generate, which these days is relatively easy. It's not easy, but generating data is one thing, but making sense of it is very complicated. And so that idea is how to triangulate this information across multiple dimensions. And so we build a number of algorithms, which, alembic is an acronym for, is the apparatus for alchemic, but is also the acronym word for algorithms for life-extending medicine using biology, informatics, and chemistry. Because that's what it is. And I'm lucky enough to work with really, really talented people. Our CTO, Alex Laslovic, was actually previously one of the lead software engineers from Facebook. So not coming from the biology.
Right.
But he came with an incredibly sophisticated software engineering skills. And on top of that, he's very, very talented. So he knows how to build infrastructures, complex systems, and build something from scratch. So I didn't want someone who comes as a bioinformatician that knows how to use the tools and just apply that. I need someone who's completely unbiased that can see this problem as a software engineer who said, okay, I need to build a tool to solve this. And he did it incredibly well. So the alembic is not just like a bunch of spreadsheets. It's actually fully user interfaced. Everybody, biologists and chemists, have their own account, they log in, and you can imagine what Alex has done is almost like a Facebook of computational discovery. So they can really work cross-functionally across data, mines data through electronic lab notes. It talks with instruments in the lab. It talks to people through Slack. Is really the medium of the company where people use it, analyze, share data, get suggestion, feedback. It's actually pretty amazing because enabled and accelerated our internal discovery process, not just as a tool, but really as, an infrastructure that we use internally or with partners outside. And so then it was like how we analyze the data, sure, but also how they are delivered, how the data are connected, how people are connected is making a big difference.
Mm-hmm.
So we use lots of course, novel math, some developed in-house, deep neural network, generative AI. I mean, all those things are tools,
Sure.
right? But they're very useful to analyze complex data in many ways. But the single cell, why single cell? I mean, it's important because many of those cell types, let's talk about senescence, for example, is one of our focus. Now, cellular senescence, what I learned over the years, and especially also working at Rubedo more in the recent years, is that, I sometimes jokes about it, but I can say that senescent cells don't exist. And I'd say provocatively, but in a way it's true. Because it's like saying a cancer cell doesn't exist. There is no such a thing as one cancer cell. There are so many, so different to define. And it's not very different from senescent cells. Maybe the field started, let's say 10, 15 years ago, thinking, how can we kill senescent cells? Like making a kind of pan-senolytic, if you will. But that's kind of in the 70s for oncology. How can we kill cancer cells? Now, after 50 years, we know we can cure cancer, but we're still heavily working on it. We have multiple modalities, many treatments, combination therapies, because it's complex. And the complexity for cellular senescence is, similar. I don't know if it's as complex, but definitely there is a high degree of heterogeneity. They're not all bad. They're pleiotropic. Some are positive and you don't want to touch. Some are pathologic, but some are consequence, some are causing. There is primary, secondary senescence, early senescence, deep senescence. So you really need to know what you have to target. And as a matter of fact, not all, let's say, senolytics or drugs targeting them work in the same way for all of them. So when we have lots of work there that, for example, the BCL2 class inhibitors like navitoclax. So this is one of the first generation of so-called senolytics. They were being repurposed from the oncology space. And they do work in vitro well on certain models, but actually work very badly or not at all in other models. Even on the same cell type, if you induce senescence, even with the same modality, we have data showing that if you use chemotherapy-induced senescence in vitro, and you use two different standards of care, cancer, to induce senescence, looking at the canonical markers like P16 or beta-gal, etc., those cells look the same. So you say, now you have senescent cells, and you may expect that then you can kill them in the same way. But when you put, let's say, navitoclax to the same cells, but induced in different ways in senescence, it can, in one case, it works, in the other doesn't. And when you look at transcriptional profile and pathways analysis and what's happening, it's very different. So they're in two different senescence states. And you need to know in a patient, which is even more complex, how do those cells look like? And they're not going to look like the same. And sometimes you cannot rely on the traditional markers that we discover mostly in vitro, which is, again, moving into the niche context is a very artificial environment that can mislead to what senescence is, maybe an extreme case or an adaptive response. But the senescent cells in vivo are really pathologic cells that might be senescent-like, but they're really causing this maladaptive response into the microenvironment, causing their other cells, stem cells, immune cells, to react and respond, but because they're resident and persistent, can really drive this progression. So it's very important to get rid of them, but there are very few, very heterogeneous. So that's why we build this strategy. So we know what to target and how to build molecules that can get rid of those in the right context and release the immune cells and stem cells, progenitors, to shift towards a pro-regenerative response that otherwise is affected by those pathologic aberrant cells that stay in the tissue. I think that's a more sober, to me, view of what senescent cells are, and that's where I really spent lots of effort in building strategies to target them very selectively And you need multiple molecules depending what you're trying to treat. So for interstitial lung diseases like lung fibrosis, COPD, pulmonary hypertensions, you have a certain biology, you might have certain targets, and so we're developing certain molecules. They're different for skin conditions such as chronic atopic dermatitis or psoriasis. Others that are generally aggravated by an aged skin. They don't resolve, they're persistent lesions. And we found that there are senescent-like cells there that are causing this maladaptive response. And that's really where we're focusing. So building a portfolio of different molecules that can have different roles and functions and beyond senescent cells, right? It doesn't have to be. So similar strategy to discover other aberrant cells that might play a role in this biological aging process.
Hence why I refer to you as, Sena Therapeutics 2.0. But yeah, no, it's again, it's a very elegant approach, and needless to say, especially the last several months, it's really been paying off for you, and you've been making some interesting collaborations along the way. And I think, as you were mentioning before, that you're not looking at low-hanging fruit, and clearly with the IDF, the IPF program, that's not low-hanging fruit at all, but nonetheless, California Institute for Regenerative Medicine and Maria Milan and team, funding you to again evaluate some of these moieties in these really unique models. Talk a little bit about the IPF program, if you would. I think this is just not just a substantial unmet medical need, but clearly the CIRM believes in you enough to fund this. It's a really nice validation that you're on an interesting path here.
Yeah, thank you. Sure. So one thing that we believe is important in degenerative lung conditions, chronic degenerative conditions, like IPF, which the field is starting to, and we also believe that probably a more appropriate name instead of idiopathic pulmonary fibrosis is really progressive pulmonary fibrosis. Is really this progression is really key. And instead of focusing on the end point or end stage of the disease, which is fibrosis, so cells that are eventually, generating collagen, which is a myofibroblast, and they generate this extracellular matrix that results in scarring, etc. Where standard of care, like there are two approved drugs in IPF, nintedanib and pirfenidone, and others in development, they focus on this, right? How to stop fibrosis, which makes sense if you think, but all those therapies showed very limited to no efficacy. Right? It's a non-disease modifying intervention. The majority of patients are not responders. When they do, they only slow the progression to a certain extent. The life expectancy depends on the type of IPF, right? There is a fast to slow, medium progression, but the average is 2 to 3 years. So similar to a cancer patient, really very limited. And maybe those drugs can give you, 6 months, maybe a year or two, but with the cost of high burden in decreased quality of life. They're very toxic. And many patients, despite their severity of their disease, they actually drop out with an average of 6 to 12 months from the treatment because they can't tolerate that anymore. So high unmet need, but very difficult indication. Now, as we started to work and study with that, and also working with KOLs and our collaborators, so we have a partnership, for example, with the Siddharth Medical Center in LA. That is one center of excellence in the world. They work on interstitial lung diseases. And for example, we work with Professor Ogobam there that has been working for like 3 decades on IPF and cellular senescence. So it's really like that role. So that was a great match in collaboration. So it came clear that it's not really the fibroblast that it's a main responsible to cause the fibrosis, but what the problem is the progression of the disease. So even if you slow down fibroblasts, unfortunately, the lung tissue continue to generate more and degenerate during this process. And as a matter of fact, typically the lower lobe of the lungs start to experience this degeneration, and slowly the disease just grows up until the entire lung fails. So the really only alternative and treatment is lung transplantation, which is very, very limited for a small number of people. But as we started to study this, we found that there are many cell types that acquire a role or aspects and flavors of senescent cells during this process. But at the top of this cascade, that drives this progression, that eventually leads to recruitment and activation of fibroblasts into myofibroblast. They start to release all this are cells that have these senescent features. And among those, there are progenitors, like stem cells and progenitors of the lung that are at the top of this cascade. And because generally those patients you need some sort of a baseline of some genetic predisposition. There are lots of familial conditions behind that. For example, mutations that affect the telomeres. TERT or sheltering or everything that makes the telomere more fragile will get shorter sooner. As a result, cells start to become senescent sooner, especially if there are cofactors, like it could be smoking, air pollution, SARS-CoV-2, a virus, right? And so while a healthy person can recover, those people, they start to build these senescent cells, especially that affecting those stem cells, they start to deplete them. So instead of having cells that can regenerate and repair, you start to accumulate, generate a progenitor of cells that are more senescent-like, that when they recruit immune cells into the niche, into this microenvironment, instead of clearing them, they participate and they respond to that, and they start to become also senescent-like. Just exponentially, let's say, escalating this process. And now you create an environment that is enriched by those pro-inflammatory, pro-fibrotic factors that then recruit fibroblasts that start to change the way they behave and create scarring, and this keep propagates. But if you remove, that's our idea, and the work with the CIRM we're doing as well, if you can target selectively those cells and stop this cascade of events, you could have a disease-modifying intervention by stopping the progression. And you might not, maybe if it's too late, not necessarily repair the scarring if you're an extended fibrotic tissue, but you can stop or slow down the progression, and that starts a lower lobe, so you preserve function of your lungs that is still remaining, and promote the healthy stem cells to repair. So it's a profoundly different approach, also because what we see that those senescent cells of the lung are really the factory of many, many pro-fibrotic factors, and therapies that have been trying to target one at a time, like an anti-TGF-beta treatment, for example, has a limited effect because then there are so many others that are still released and drive this fibrosis. Removing those senescent cells is just removing at once the source of so many pro-fibrotic factors that really drop this baseline and enable immune cells and stem cells to change their behavior.
Really, really fascinating. And again, bringing this niche thinking in is just so important. As you plan your strategy, it's really fascinating work. Marco, along those lines, could you say a few words about RLS 1496? Because I know that was also recently an announcement that here we're talking about dermatology, but again, a broad swath of possibilities, everything from atopic dermatitis to psoriasis. And having worked in this space in previous career, it's very large, and love to hear about your plans to get to IND enabling through the IND enabling studies, and I believe first in human next year. So that's a rather rapid timeline, but say a few words about that one too, if you would.
Yeah, it's true. That was a rapid pipeline. We actually started this program in dermatology relatively recently, after raising our Series A, which by the way we never announced, but that was at late 2021. Back then, we only had the program in lung because we were smaller and more focused. When we started to build on the platform and explore other programs, dermatology was one for a number of reasons, and went really quickly from zero to target discovery to hit to lead up and nomination of a development candidate in about a year and a half. So it was a very also good validation of the platform and the strategy. This is a novel target, which is still unannounced, but is a first-in-class new molecule that target those classes of senescent cells of the skin that we found that we think they're like similarly to what I'm saying in the lung, right? The idea here is those. So we identified about dozens of indications so far. Now we definitely selected chronic psoriasis and chronic atopic dermatitis, but there are others, and we plan to test them, several of them, in a trial. So doing a basket trial, and instead of focusing on one, because I will give us really instead of spending more time doing preclinical studies, we're going to test this in a clinical study. It's really a clinical experimentation, because we really rely heavily on clinical human data and clinical work. In my experience, animal models are really poorly recapitulating. Dogs, pigs, rats, mice, they're limited. And you get the perception that you might be doing something that is relevant, and 99% of the case is not.
Right.
So it is informative, but to a very limited extent. So, but those conditions can be very different. Like in one case, an autoimmune; in the other case, could be driven by a agent, etc. But what they have in common? They have in common this common denominator is that they have persistent skin lesions. They are characterized by chronic inflammation, and that's where we see that those lesions have the potential to resolve. And sometimes it takes months, years, and sometimes they don't. They degenerate. They could degenerate in skin fibrosis, for example, or things like that. Standard of care, like first line of treatment, are typically steroids, topical steroids, and they work. But in the very acute phase, when those conditions become chronic, they actually make things worse. And there is a rebound effect and sensitization, etc. And what we see is really there is a major difference. In the acute phase, you don't really see those senescent-like cells. They start to build up later, and when they start to accumulate, they shift. Even those immune cells are there, but they can't heal anymore. They respond to this new environment. And the struggle to clear those lesions. And so once again, try to target a factor at a time has limited effect. So even biologics, etc., it works in subset of patients, but it remains for many of those conditions and highly unmet medical need, with a lack of real disease-modifying intervention in many cases. So we think that we have an opportunity for a disease-modifying intervention that could be first in line. So an alternative or a follow-up to steroids prior to start to do biologics and injections and oral, etc. Because those cells can really, once you clear them, you enable the immune cells that are there to do their job and clear those conditions. That's what our nominated candidate for the derm program is targeting those. And we start with topical, but we also have oral formulations and really to go after more systemic conditions. So the topical program, which is an attractive, interesting entry point into large markets, etc., will also be validation for the target and the specific strategy to then go systemic for other conditions, for example, but also look at secondary endpoints that might look at how they impact the skin aging, for example, for other use and applications. So we really think it's a very way to fast track the clinical development, validate the target, and validate the strategy with the initial program in dermatology. But our goal is not to be or become a dermatological company. It's only one of many directions.
Absolutely. Yeah. But no, it's again, it speaks to the diversification of the portfolio, and again, it's pretty impressive how far you've come with that one as well. Mark, I gotta ask, because I'm sitting here. Obviously, this episode I'm talking to you as CEO of Rubedo, but as I mentioned in the bio, you're also the founder of Turn, which is a reprogramming company, the Wetware Concepts company. You've been involved in all this interesting stuff with electrical stimulation and peripheral nerves. I mean, I obviously if I showed up tomorrow and I gave you a couple $1 billion, right, you could invest $1 billion into each of these enterprises and develop individual companies to their potential. But obviously, one steps back and sees the connection between senescence and reprogramming and neuralization, and they all feed into sort of the big picture of complex regeneration and rejuvenation that we see other animals in the biologic kingdom capable of wielding in synergy to do things like regenerate arms and entire organs and things like that. You got sort of a big picture dream for all this. If I did show up with a few $1 billion for you, where some of this stuff becomes integrated together into a single entity, because it seems like it all fits a nice little puzzle.
It does. Well, so please show up with a few billions if you can. That would be great.
Not crypto either.
But no, certainly. I mean, again, you're absolutely right. All those things kind of come together. I mean, where I did my first bet, recently is in, on Rubedo and Turn. If I had to simplify the vision, is Rubedo is focusing removing or managing the bad cells that are resulting from this biological aging process. You cannot live with them. If you have them, then things start to go bad. So you need to get rid of them. But you also have the good cells that tends to become bad over time. And that's what Turns aims for by using a transient epigenetic reprogramming. And we're using an RNA-based technology to deliver those factors and control this process transiently. You protect the good cells, you maintain the regenerative function and prevent them to become pathologic, to become senescent and many others. So my aspirational goal is this two types of strategies in the future can be used synergistically together in combination to really advance to the next step of truly rejuvenative therapies. But first, we need to start with clearer defined indication with clear endpoints, measurable within a certain time frame, and validate this. So it's a stepwise process. But I always, sometimes if the impression is, oh, you're like falling into the rabbit hole of clinical indications, no, it's part of a clear plan and the path that you have to build strategically to make a company sustainable, to advance the science in a stepwise process, but always aligned with the ultimate mission, which is, changing the trajectory of the biological aging. But, there is still a long way. So we need to do one step at a time.
Absolutely. But yeah, nonetheless, it's inspiring to see these different baskets that you put together, because I think it ultimately all leads in one very important direction. So that it's exciting. Mark, while we have you, and get back on the alchemical theme for a second, I noticed that the mission statement of the Fadon Institute is transmuting awareness of longevity and aging sciences. So I caught that alchemical reference there as well. But talk a little bit. Obviously, this is another hat you wear in terms of this think tank and obviously awareness and the appropriate awareness, let's say, of what's going on in this industry is so extremely important. Talk about why you set up this organization, a little bit of what you're up to. I know you have some conferences that you work on. I see Eric Fadon is involved with you. We had on the show a couple months ago. Talk a little bit about what you're doing there, if you would.
Yeah. So the Fadon Institute, and well, the name comes from the Greek philosophy, by the way, and also from Plato, where Phaedon is really means like bringing light, shining light from the inside to outside, and typically is associated with like bringing light to the mind and the soul, in a way, like, and I was associated with this, let's say, immortality of not just the mind, but of the soul. Yeah. But the reason is why the Fadon Institute, and there are so many organizations already working on awareness on longevity, etc. And maybe that's exactly the reason, right? I found myself, especially a year ago when I decided to take some action, that frustrated when the term longevity or aging is now becoming extremely diluted and doesn't really mean much anymore, is unfortunately often associated with pseudoscience or snake oil. And the problem is that everything is mix and match together, right? It's such a big field, and we need to be careful how to navigate that. Right. And even in large conferences, even my favorite ones, where I love to go and speak, but still you never go deep because it's all together. So you hear about everything, but you never go deep enough. And so what I realized that talking even with people that maybe are not, KOLs or experts, but still that are familiar with the space, that there is lots of misconception. And sometimes you hear the wrong things or old things. And there is so much confusion. And I think this is not good because we need clarity. We need to raise the bar high in terms of rigor, scientific rigor, and clinical rigor, etc. And I feel like people don't know, well, what are the right biomarkers? What are the right indications? How is a good clinical trial? I mean, is senescence good or bad? Oh, the first trials didn't show efficacy, so let's move to the next thing. I say, wait, wait a minute. That's not how it works, right? Experiments are primed to fail. That's how we learn. All the fields go through this process. We are learning a lot. Actually, the field is progressing very significantly. Just to give an example of cellular senescence. So it's actually, we need to make clarity, but we need that focus. And I realize there is really not a forum for that. And it's a risk for all the other fields under the longevity hat, longevity medicine or science. So epigenetic reprogramming, mitochondrial-based therapies, etc. So I felt, well, so if I'm frustrated, maybe I should do something. And that's why with other KOLs, we founded this scientific society. This is what it kind of is, kind of inspired by like the Gordon Society. Sure. But focus on the longevity space, but differentiating in different areas. So we can bring together all the stakeholders and that people that are working and making difference and putting effort to advance that specific areas, which is very diversified by others. So yeah, it's broadly speaking aging, but it's actually very, very different what people are doing. It's like in biology, in a way. So that's the idea of this think tank and all the initiatives and events around it. So the first event, like you mentioned, is this Therapeutic Summit, which will be at the Buck Institute this coming November 7th, 8th, which I invite everybody listening to join. If you're interested, it will be, I think, a unique first experiment, but also unique initiative because we put together everyone that we could, and I think really we did a good job there, of people that are working in the cellular senescence science and the xenotherapeutic development, cross-functionally. So there are academics, clinicians, industry, all the early stage companies working in these developing therapies like Rubedo, but also late-stage companies that are maybe already in the clinic, and big global pharma.
Mm-hmm.
For example, we have a panel of from big pharma where we have like the CSO of Novartis will be there. We have VPs from Eli Lilly, from Boehringer Ingelheim, Ono Pharmaceuticals, etc., to get their perspective and say, okay, what are bottlenecks, opportunities, risks? We partner up with a consortium of biomarkers of aging to discuss, how to consider relevant biomarkers for a clinical trial, how to run a clinical trial. Let's not reinvent the wheel, but let's work together.
Yep.
NPG, Nature Aging, is, for example, one of the sponsors. So we're going to publish the, let's say, outcome, the take-home message we want with the recommendations. The really ultimate goal is to create a forum for people who are in there, an opportunity for people who wants to really learn what's going on in that field, what matters, what's important, not to be exposed to that and contribute, and to help each other. Because I don't see any competitor. I think the field is very small. We need much more, and everybody is a collaborator. And my goal is help everyone to succeed, because we need successful stories to build more confidence and advance the field. Errors will only push back things by years sometimes. So I really want to help to create these synergies, create winners, and make clarity. And that's what the Fadon Institute is. And the Xenotherapeutic Summit is the first initiative. But next year, we plan to do other similar events and other initiatives on the different areas and fields somewhat around this aging biology field.
Mm-hmm. And it's another wonderful initiative. I'm glad you're running it, and we'll put the links to the upcoming conference in the bio of the show. One last thing, Mark, while I have you, aside from the Phaedon initiatives, anything else? I know you were recently, I think, at ARD in Copenhagen. Anything else for 2023 as we head into 2024 that we should know about in terms of your public-facing initiatives, places that we can run into you, meet you, listen to you talk about things you're doing, other conferences you're going to be at, talks? Anything I missed, please take the floor.
Well, let me see. I will be in Minneapolis for the International Cellular Senescence Society conference. So people will be there. It's purely very academic. So compared to the Xenotherapeutic Summit, and many people there will also be speaking at the summit. A few weeks later. That's at the end of October, but that's one. I have a few events, but I will be in Riyadh for the next event organized by Evolution Foundation in the Saudi Arabia, which is another important event. I think they're doing a great job there to build and support the field from the Gulf area and that part of the region. There is a longevity summit at the back in December. That is happening again. I think these are some of the probably good events up and coming over the next few months where I will be around and always happy to meet people. So if anyone then is interested, I'm always available. So I invite people to reach out through LinkedIn or through the website, and happy always to connect.
Excellent. Anyway, again, we will put those links in the bio of the show. Amazing work, Marco. I'm a big fan for a while now, rooting you on as you continue to build this amazing structure and strategy. For everybody that's going to be listening to this particular episode of our show across the various podcast networks or watching on our YouTube channel, again, you've been spending time with Dr. Marco Quarta, co-founder, chief executive officer, Rubedo Life Sciences, doing really amazing things at the intersection of aging, stem cells, regenerative medicine, rejuvenation, and everything in between. Marco, I want to thank you for taking the time out of your schedule to come talk to us for a little while, to educate us. Obviously, thank you for what you do. And as we like to say here on our show, thanks for helping to create a better tomorrow for everybody out there. Really a great story and rooting you on.
Well, thank you for your kind words, and thank you for all the great work that you're doing, also to support the field, the communication around it. I think your podcast is one of the definitely best medium out there. So I'm really, really happy to contribute with this today. And yeah, I'll look forward to continue discussions. But thank you for having me here today.
Great having you.